Hello, everyone, and welcome to our next Eco-Evo seminar of this Wednesday. So my name is Wouter van der Bijl. I'm a postdoc with Judith Menk at UBC. And before I introduce today's speaker, I want to briefly mention our schedule to you. So next week, August 19th, we actually don't have a seminar planned, and we'll be taking 1 week off. But no worries, because the week after we'll be back again. And then we'll have Simon Zandwe. He'll be speaking about something, I don't have an official title, but my guess is he will be talking about genome duplication and Atlantic salmon. If that's not true, I apologize, but that's my guess at least. Okay, so without further ado, I'd like to introduce today's speaker, who's Professor Martha Muñoz. Martha did her PhD with Jonathan Losos at Harvard, working on the famous Anolis lizard system. She then moved to Australia to become a postdoc at ANU, after which she returned to the US for a postdoc at Duke University. She first became a PI in 2017, when she became assistant professor at Virginia Tech, but she moved to Yale last year. In 2017, Martha won the Young Investigator Award of the American Society of Naturalists. And Martha's work often combines physiology and biomechanics with evolutionary biology, which I think is a quite interesting combination. But perhaps the best overarching theme to describe her work is to understand why there's variation in evolutionary rates across the tree of life. And with that, Martha, the stream is yours.
Thank you so much, Wouter, for that wonderful introduction. And thank you to all of the organizers for inviting me to speak. I'm delighted to be able to share some of my work with you today. So to get started, as Wouter intimated, I'm going to start with an observation in evolution, which is that it proceeds unevenly. And by this, I mean that some traits and lineages evolve much more quickly than others, and others, in contrast, can remain inert for millions of years, or nearly so for millions of years. To give an example, consider that that lizard genitalia evolve much more quickly than other aspects of the phenotype, to the point, and this is from an actual press release, that lizard penises can be inferred to be evolving at super speed. Just as exciting is that certain aspects of the fish mandible evolve up to 30 times faster than others. Now, keep in mind that these are integrated structures that need to work together to accomplish fitness-related functions, and yet the rates of evolution can be so decoupled from each other and evolve at vastly different rates. So why is this? Well, what I'm suggesting, or what we're going to explore today, is that there are intrinsic and extrinsic factors to organisms that allow evolution in some cases to proceed exceptionally rapidly, facilitate it, whereas in others, evolution can proceed much more slowly or even be stopped dead in its tracks. So why is this? Why do some traits get to go on evolution's Autobahn, whereas others are stuck in New York City traffic at rush hour? Well, I explore this phenomenon by examining one of evolution's key architects, which is behavior. Behavior allows organisms to exploit new resources or enter new environments. Behavior is one of the key architects of evolution. By exploiting new environments, organisms can expose themselves to natural selection, in turn accelerating it, or conversely, they can shield themselves from selection, effectively stopping it dead in its tracks. And I usually examine the role of behavior in evolution through the lens of physiology, specifically with respect to how organisms interact with their thermal environments. And I do this largely in ectotherms, like lizards, but of course, there are many other organisms that are ectotherms. In fact, most animals are ectotherms. In ectotherms like lizards, the ability to perform a task such as sprinting is contingent on body temperature, such that it's optimized over a relatively narrow range of temperatures, and then performance decreases at higher and lower temperatures until the organism is immobilized. Now, my approach is to dissect this curve into its component features. So, for example, this curve has critical thermal limits, temperatures above and below which organisms can no longer move. This curve has a maximum performance, a temperature at which performance is optimized, and a preferred thermal range, a range of temperatures over which performance is optimal or nearly so. I dissect this curve into all of its component features and then examine the factors shaping their evolution. And as Wouter intimated, I do this primarily, though not exclusively, with Anolis lizards. So for the uninitiated, anoles are an extremely diverse lineage of reptiles from the tropical and subtropical regions of the Western Hemisphere. There are about 400 species or so currently described, with more pretty much every other week. And they're exceptionally diverse in the Caribbean, where more than 150 species of anoles can be found. And of course, they are colorful and charismatic organisms to work with. They have these extensible throat fans termed dewlaps, which they use in social communication, territorial defense, and they're just these lovely, colorful organisms to work with. But today, I'm going to instead focus on my favorite type of anole, the LBL, the little brown lizard, and I will be focusing specifically on a lineage of lizards from the Caribbean island of Hispaniola. This lineage, termed the Cybotoids, is comprised of Anolis cybotes, this lizard featured here, and its close relatives. And what this lineage of mostly brown lizards lack in oranges and blues and greens, I promise they make up for in physiological badassery. So to give a little outline of where I hope to go today, I'm going to start with some evidence for the arresting role of behavior in evolution, and then switch gears and show how the same behavior, in fact, might have both arresting and accelerating impacts on evolution. Then I hope to take a step back and simply ask, why do we care about the role of behavior in evolution? Does it matter for important, crises like global climate change? And wrap up hopefully with some new perspectives on old ideas on the role of behavior in evolution, and give a little hint of what's to come. So when I started working with anoles, I was especially fascinated by the island of Hispaniola. This is an island that's dissected into two main mountain chains, the Cordillera Central in the east and the Sierra de Bauruco in the west. And this island harbors the highest peak in the Caribbean. You can go 3,000 meters above sea level and still be on a Caribbean island if you're on Hispaniola. Now, near sea level, Hispaniola looks a lot like what you might expect from a tropical island. Lots of lush, warm, mesic broadleaf forest. The tropical anoles that get to live at low elevation here live a pretty cush and comfortable life. As you start to go up in Hispaniola, and I mean up in Hispaniola, the habitat starts to change dramatically. And to me, it starts to look more like a moonscape by comparison. I did my PhD in Massachusetts, and many times when I was in the field in the Dominican Republic, I had to remind myself that I wasn't, hiking in New Hampshire, for example. That's how different it feels. It's comprised instead of this really widespread monodominant pine forest flanked by these bunch grasses and savannas and rocky outcrops. And it gets cold up there, very, very cold. As compared to the low elevation sites on this island, those high elevation environments can be up to 20 degrees Celsius colder, which for a tropical lizard, that is a very, very cold environment. It's not uncommon, even in summer, to observe frost at the higher elevations of Hispaniola. This is me. When I first did when I did my first expedition in Hispaniola, I was actually woefully underprepared for just how cold it gets, to the point that I sheltered myself, even in summer, using spare suitcases and all of the clothing that I had in on my person to keep warm. But I wasn't alone up there. I had excellent company. On the highest elevations of Hispaniola, you get anoles. So in the eastern mountains, the Cordillera Central, you can see Anolis shrivi in really high elevation environments. And in the Sierra de Bauruco, those western mountains, you can see Anolis armori. And these high elevation anoles are part of a larger lineage, the Cybotoid Anoles, which is collectively found across the entire island of Hispaniola. But not all species in this lineage are distributed, across all elevations. There are two species, Anolis whitemani and Anolis longitubialis, which are essentially restricted to low elevation environments near sea level. There are two species, Anolis strami and marcanoi, which creep into mid-elevation habitats, 900 to 1,000 meters. There's one species, Anolis cybotes, which is essentially found island-wide up until about 1800 meters, when it starts to get replaced by Anolis armori in the western mountains and Anolis shrivi in the eastern mountains. And the boundary separating Anolis cybotes from these two montane endemics is usually the transition from the broadleaf forests, where Anolis cybotes can be found on the edge of those forests, into the high elevation monodominant pine forest stands where you only get those montane endemics. So I was just purely fascinated with the diversity of this, tropical lineage of anoles found in this wide diversity of thermal habitats. So my first step was simply, it was curiosity driven. I just wanted to know how, if at all, these tropical lizards were compensating physiologically for their thermal environments, for such different thermal environments. And I did that by focusing on two key aspects of the thermal performance curve, the critical thermal minimum and the critical thermal maximum. Now, the critical thermal minimum and maximum simply refer to the temperatures at which lizards lose the ability to right themselves. They're sort of the critical limits of locomotion. And they're measured by flipping lizards on, systematically cooling and heating lizards and flipping them on their back and determining the temperature at which they lose the ability to right themselves. Those are the critical thermal limits. And that's exactly what I'm demonstrating in this photo over here using extremely sophisticated equipment of a heat lamp and a bucket. So that's how we do the experiments. So what I did, along with my incredible team of undergraduates who accompanied me on this trip, was scour the island for as many populations of these different lizard species as we could find and just measure their critical thermal limits. So here's what we observed. I'm going to start first with CT min, or cold tolerance. On the x-axis of this plot is elevation, from low elevation on the left to high elevation on the right. The critical thermal minimum is on the y-axis. A lower CT min indicates an organism that is more cold tolerant. It was able to withstand a cooler temperature before losing the righting response. And each point is going to show you the population mean plus the standard deviation, and the color of the point is going to refer to the different species, the species from which the data were gathered. And what we found was, perhaps unsurprisingly, was a very strong clinal relationship between cold tolerance and elevation, such that these montane lizards were up to 7 degrees Celsius more cold tolerant than their low elevation counterparts. This makes sense, but even for a lineage, these are all highly, very closely related lizards. I was struck by how dramatic this change in CT min was across elevation. Okay, what about CT max? So the same plot as before, elevation is on the x-axis, and in this case, the critical thermal maximum, CT max, is on the y-axis. A higher value indicates a population of lizards that is more heat tolerant in this case. Here's what we found. Absolutely no relationship whatsoever between heat tolerance and elevation, and about half as much within population variation for heat tolerance than for cold tolerance. And I found this result a little more challenging to digest, a little harder to explain. So I decided to take a step back and just more deeply explore the temperatures that are actually available to organisms in the habitats that they occupy. So what I did was visit 4 sites at different elevations, 45 meters, 700, 1600, and 2450, and just set out a lot of sensors in each of these habitats to record temperatures over the course of a single day, and over a single day, separating nighttime and daytime temperatures. I'm going to show you first the temperatures that I measured at night. On this plot, time is on the x-axis from sundown to sun up, temperatures on the y-axis, and the color is going to correspond to the different sites from which I measured temperatures. So and secondly, very little, if any, overlap in temperatures among sites. So in light of these results, we can consider temperature to be elevationally structured in Hispaniola into distinct thermal zones, essentially. that go from warmer temperatures at lower elevation to cooler at high elevation. And perhaps unsurprisingly, the phenotypic trait, in this case, cold tolerance, exhibits a clinal relationship that seems to sort of match the available temperatures at night in their habitats. Nighttime being when temperatures are coolest, and therefore when traits like CT-MIN should be under stronger selection. Let's do the exact same exercise, but with daytime temperatures. So in this case, temperature is again on the y-axis, but now we're going from sun up to sundown. And here's what we observe for those same 4 sites. A lot more thermal variation during the day. In fact, whereas night times you had very compressed thermal variation, 5 to 10 degrees Celsius, during the day, that expands dramatically to 30 to 40 degrees Celsius at any given site. And importantly, they're not just, these sites are not just more thermally variable during the day, but a lot of that thermal variation is shared across elevation. Perhaps it doesn't get quite so warm at at high elevation, or vice versa, but overall you can see clearly that the temperatures overlap a lot among these sites during the day. Correspondingly, you could consider Hispaniola during the day to look a lot more like this. There's a lot more thermal variation, and that variation is shared across elevation. If organisms' phenotypes, in this case heat tolerance, were to more closely match the environment, we might expect that there'd be a lot of within population variation in CT max and a clinal relationship with CT max across elevation. And yet that's not what we observe at all. In fact, the data for CT max look a lot more like this: very little within population variation and no relationship across elevation. So what gives? There's a piece that was missing from this puzzle. So Wouter, can you tell me something interesting, not to pick on you, that's going on in this image that this lizard might be doing?
It's finding shade.
It might, yeah, it could be looking for shade. Yulia, do you observe any interesting behaviors or something going on?
Maybe its feet are not touching the leaf.
That's right, exactly. So if you look at its hind foot, it's elevated off the surface of the hot leaf. And do you see the toes on the forelimbs, how those are curled back? It's behaviorally reducing its contact with the hot leaf. It's using behavioral thermal regulation to essentially modify its environment. So, and this was the piece that was missing. So to take a step back, the idea of thermal regulation may seem intuitive and obvious to you and me, but as a concept, it's actually been formalized for less than a century. It was formalized in the 1940s by 2 gentlemen, Bob Cowles and Charles Bogert, who spent many years running around the American Southwest and measuring temperatures from desert lizards. And they were struck by how stable and cool lizard body temperatures were relative to the really hot desert environments that they occupied. And that's how the concept was originally formalized. So to explain the idea of thermal regulation, let's take a bird's eye view of a thermal habitat. Perhaps deep in the forest, the temperatures are too cool for lizards. And maybe out in the open, they could fry and it's just too hot for them. Maybe it's really only those trees that sort of flank the periphery between open and closed habitats that are thermally optimal for lizards. Let's go ahead and make those thermally optimal patches, an Evo Eco Seminars shade of teal. And let's assume that a lizard were to randomly shuttle between all available habitat, all available microclimates in its habitat. Such a thermal conforming lizard would indeed experience, the full range of temperatures available in its environment. In contrast, however, if a thermal regulating lizard were to simply preferentially shuttle between thermally optimal patches, it would in practice only experience a relatively narrow subset of the available temperatures to it. In fact, the thermal environment of such an organism might look more like this. And so by thermal regulating, organisms can effectively buffer themselves from environmental variation from temperatures that are too warm or too cool for them. And that's precisely what these Cybotoid Anoles are doing. When we go back to the same exact sites from which we measured CT max and CT min, and now just gather field active body temperature, what we observe is that it really only drops about 1.5 degrees Celsius across 2.5 kilometers of elevation. To put this into context, if those same lizards were thermal conforming, body temperature should have dropped by about 10 degrees Celsius. So they're clearly thermal regulating, which helps explain why heat tolerance remains so stable. So what they're doing is transforming a lot of the thermal availability and compressing it into the thermally optimal range for them. But we can take this one step further still. So after the concept was formalized, one of the architects of the idea, Charles Bogert, came back a few years later and gave it a bit more thought. And he said, wait a minute, by thermoregulating, organisms aren't just modifying the temperatures that they experience in their environments. In so doing, they're effectively altering natural selection. They're by shielding themselves from environmental variation, they can actually buffer themselves from selection on physiology. And he hypothesized that the traits that could be buffered from selection should evolve more slowly than the traits that cannot be buffered from selection. In honor of Charles Bogert's pioneering ideas, we've come to call this the Bogert Effect, behavior buffering organisms from selection and slowing evolution, or you may also know it as behavioral inertia. So based on this concept, we could hypothesize that all of that thermal heterogeneity during the day gives organisms the opportunity to behaviorally thermoregulate, and therefore the upper features of the thermal performance curve, like CT max, might evolve more slowly than CT min, a trait that is under selection when temperatures are actually quite homogeneous, which sort of precludes the ability to behaviorally thermoregulate. That's exactly what's going on. CT min not only evolves faster than heat tolerance and body temperature, in the case of comparison to heat tolerance, it evolves an order of magnitude faster. So this idea is that behavior isn't just, something that organisms do to interact with their environment. It's also has an evolutionary signature. And in the case of behavioral thermoregulation, that signature is a slower rate of evolution. So one thing that struck me, however, I couldn't quite marry the idea in my mind is, why are these Montane lizards going through the ordeal of behaviorally adjusting for their cold environments? Why not just adjust your physiology to the cold environment that you occupy? So my next step was simply to gather as many heat tolerance data as I could find in squamate reptiles to ask whether, is it the case that CT max is just evolutionarily inert for other reasons, for example. And I was able to gather data for about 270 reptile species and found more than 25 degrees Celsius variation in the measured CT max, which is extraordinarily substantial. So it's not the case that CT max can't evolve. When we zoom into anoles, we find, about 20 degrees Celsius variation in CT max. So it's certainly not the case that CT max can't evolve. It certainly does. But instead, what I think is occurring is that these Montane lizards are capitalizing on the physiological benefits of a higher core temperature. Now, due to some pretty universal biochemical principles, when organisms perform a task at a higher temperature, up until a certain point, they generally are able to do so at a higher maximum performance. So okay, so far, I think I've, hammered in the arresting role of behavioral thermoregulation in evolution. But our next question, or the next thing we started to wonder, was, to what extent are these data, generalizable? Is this, a Cybotoid phenomenon? Is this something that you only observe in Hispaniola for reasons we haven't yet quantified? Is this an anole phenomenon? Is it an island phenomenon? At what level does this Bogert effect signature fall apart? And so the next thing that I wanted to do was sort of expand this idea, this analysis, a bit more widely, and just look at the anole radiation in general. And the work I'm about to describe was led by Giancarlo Salazar, who is currently a graduate student at WashU, but at the time he was an undergraduate in my lab, and he performed the Herculean task of gathering every datum that was ever recorded for body temperature, heat tolerance, or cold tolerance in anoles, across multiple studies and including several experiments of his own. And what we ended up with was a data set of about 115 species. And our goal was simply to ask, does this Bogert effect still play out at deeper phylogenetic scales? So our first step in this process was to test whether the signature of thermoregulation differs between island and mainland anoles. So I'm going to show you here on this plot, mean annual temperatures on the x-axis and mean body temperatures on the y-axis. And mean annual temperature in this case is just meant to be, a relatively crude proxy of the thermal environment that these different anole species occupy. Island species are going to be shown with orange dots, and mainland species are going to be shown with blue dots. And what we found was a very clear, stronger signature of thermoregulation in island species when compared to mainland species. Now, body temperature data are inherently noisy. You're subject to weather and a whole other slew of conditions. And yet I was personally struck by how high and relatively stable island body temperatures tend to be, and how relatively neatly mainland anole body temperatures seem to, pretty much track their thermal environment. So based on this, we inferred that the signature of thermoregulation is pretty clearly stronger in island environments in general. And therefore, it might be the case, as a follow-up, that the Bogert effect, that arresting effect of thermoregulation, might be stronger in island environments than on the mainland. And if so, we had a couple of hypotheses to test. First, we would, of course, infer that there's a slower rate of heat tolerance evolution on islands. Because of the performance benefits of doing things at a higher core temperature, we would infer that if they're thermoregulating to a higher core temperature, we might also observe a higher evolutionary optimal temperature for heat tolerance in island lizards. And as somewhat of a sanity check and a control on this entire schema, cold tolerance, we hypothesize, shouldn't really differ between island and mainland lizards if it's thermoregulation that's, running the show here. So in order to test these ideas, we wanted to compare the evolutionary dynamics of heat and cold tolerance among mainland and island lizards. And in order to do that, we fitted a series of evolutionary models to each of the traits. And I'm going to walk you through some of the models. The simplest model is a simple single-rate Brownian motion model in which the differences in heat tolerance or cold tolerance on species are simply proportionate to branch length or time. A slightly more complex Brownian motion model allows the rate of evolution to differ between mainland and island species. The other three models are adaptive or Ornstein-Uhlenbeck models of trait evolution, in which there's assumed to be an optimal trait value and a rubber band parameter attracting organisms to that evolutionary optimum. The simplest OU model was one in which there was a global adaptive peak for both mainland and island lizards, and a slightly more complex model simply allowed the optimal trait value, or theta, to vary between mainland and island lizards. So in short, there were 5 models that varied in complexity, and we ran these analyses over 500 stochastic character maps for each of the traits, and the overwhelming support was different between heat and cold tolerance. For cold tolerance, we really didn't detect any major differences between mainland and island lizards. The best model that explained the data was a single-rate Brownian motion model. However, in contrast, heat tolerance definitely differs between mainland and island anoles, and they differ. The evolutionary dynamics of heat tolerance differs both in the optimal trait value and in the evolutionary rate. So the question, of course, is, does it vary in a way that's consistent with the Bogert effect being stronger on islands? So if that's the case, we would expect a slower evolutionary rate on islands and a higher evolutionary optimal trait value on islands. And that's precisely what we observe. Heat tolerance evolves 3 to 4 times more slowly on islands, and it evolves to about a 4 degrees Celsius warmer heat tolerance than in the mainland. So they're evolving to tolerate higher body temperatures. If you know anything about anoles, this result is actually kind of odd. And it's a bit weird because, if anything, anoles, especially island anoles, are a paragon of rapid evolution. And if you were to look at morphology, that's precisely what you would observe. These are 3 traits: body size, tail length, and head length, that are highly correlated with ecomorphology, this very critical aspect of the adaptive radiation of anoles. And of course, the evolutionary rate is faster for island lizards than mainland anoles. And the reason for this is ecological opportunity. So these island lizards washed up on these islands and found themselves essentially released from their ancestral competitors and predators, and were able to rapidly diversify in morphology to occupy different structural niches in their habitats. And yet, we have this slightly bizarre result of slower evolution on islands. So what gives? Well, we explored a couple of options. Maybe islands are just warmer, or climatic niches turn over faster on continents, or at a finer scale, the available thermal habitat differs. To the extent that we were able to assess these ideas, we didn't find any support. We did find one major difference between anoles. So between mainland and island anoles. So for some context, my first expedition to work with anoles was in Costa Rica, which is a mainland habitat. And I observed anoles. I did behavioral trials there. And what I mostly saw was them doing exactly what you see this anole doing, which is nothing much, honestly, hiding and attempting to look as cryptic as possible. And I thought, based on my limited experience, that anoles just simply didn't move around their habitats very much. And then I started working on island anoles and found that they spend most of their time, in fact, moving around their habitats, displaying, foraging, defending territories, and in my opinion, being as visually loud as possible and making themselves extremely conspicuous. And these movement rates are, of course, the signature of ecological opportunity, this release from predators. And yet the ability to more freely move around your habitat doesn't just affect structural microhabitat use and ecomorphological evolution, it affects thermal regulation. Because whether or not organisms thermoregulate is a balance between the benefits, such as a performance advantage, and its costs, namely exposure to predators and competitors, which is precisely what these island lizards have been released from. When we put this all together, we come to the somewhat surprising conclusion that ecological opportunity, this feature that has almost been universally associated with faster evolution, can actually indirectly slow evolution by reducing the costs of thermal regulation in island environments. Okay, so I think I've beat the drum quite a bit about the arresting force of evolution, behavioral inertia, and the Bogert effect. And that is, there's validity to those ideas. They've been around for quite some time. But the opposite idea to this has also been around for just as long, and if anything, might even be considered the classic view of the role of behavior in evolution. So led in part by many figures, but chief among them, I would say, was Ernst Mayr, is this idea that behavior should drive evolution. So behavior allows organisms to exploit new resources in their environment or to enter new environments altogether, and in so doing, expose themselves to natural selection and effectively drive evolution. Now, behavioral inertia, or the Bogert effect, and behavioral drive are not mutually exclusive ideas by any stretch. They're both, equally valid, but they've been largely studied independently. One might gather some data for a trait in a lineage and test whether the evolution of that trait is consistent with either of these ideas. I'd like to propose a slightly different concept, which is that behavioral drive and inertia might be mechanistically linked, because niches are multidimensional and the resources that organisms use can often span multiple niche dimensions. So it may be the case that whatever organisms are doing to thermoregulate at high elevation might involve a switch in resource use, which might have actually a contrasting evolutionary impact on some other aspect of the phenotype. This is the idea that I wanted to test going into this study. So take a moment. In order to test that idea, I really needed to know what the heck were these mountain lizards doing to so exquisitely thermoregulate in their super cold environments? What on the ground are they actually doing? So what I did was visit low and high elevation sites from both the Cordillera Central and the Sierra de Bauruco to explore exactly that. These are the low elevation sites that I visited, your typical, mesic broadleaf forest, tropical broadleaf forest. And then at high elevation, again, we're in these monodominant pine forests surrounded by rocky outcrops and savannas, very different habitat, mean annual temperature of about 10 to 11 degrees Celsius. In order to mechanistically understand whether and how organisms are thermoregulating, you need three pieces of data. First, you need some understanding of the available temperatures to organisms. What could they theoretically achieve in each of their habitats? Then you need to know what are their actual body temperatures, what subset of the environment, if any, are they capitalizing upon? And finally, you also need to know, in the absence of ecological constraints, what thermal range are they theoretically targeting? What's their preferred thermal conditions? So to gather the available thermal environmental data, what I did was deploy these copper lizards. These are literally electroformed copper in the shape of a lizard, inside of which we've embedded these eye button devices. And electroformed copper heats up and cools off at a rate comparable to live lizards. So when you deploy these in large numbers, you're able to create a null distribution of available temperatures to the lizards. Now, these anoles are in the are microhabitat specialists that we term trunk ground anoles. So they are most often found on tree trunks and on boulders. And so those are the perches onto which I deployed my devices. And they were in each of those 4 habitats, continuously sampling every 10 minutes over the course of 6 days in each habitat. At the same time that those sensors were measuring temperature, we were out in those habitats measuring body temperatures from live lizards. So this is me and my undergraduates doing what, of course, is the funnest part of the study, which is catching the lizards and measuring their body temperatures. And then we brought a subset of those lizards back to the lab to measure their preferred thermal conditions. So we put them in a thermal gradient and allowed to choose where to sit. And from this, we were able to extract their preferred thermal conditions. So to give you an idea of what the data look like, you're going to see some environmental data, right? I've created some dummy data here to show you what they look like. This is a histogram. If the body temperature data sort of, pretty cleanly overlap in mean and range with the available thermal conditions, we would infer that those lizards are thermoconformers. If, in contrast, the mean and the range of temperatures is quite different from that which is available in the habitat, we would instead infer that the lizards are thermoregulators. So here are the data. I'm going to start with low elevation first, with the western mountains on the left and the eastern on the right. And I'm going to show you the temperatures I measured on tree trunks, boulders, and then lizards. So here are the temperatures I measured on boulders. Here are the temperatures I measured on trees. Here are the lizard body temperatures. So, Yulia, thermoconformers or thermoregulators?
Thermoconformers?
Yes, very much thermoconformers. These lizards are pretty passive with respect to their thermal environment, but there's a reason why, and it's because their preferred range pretty much overlaps with the most available thermal, temperatures in their environment. So put differently, these low elevation lizards aren't precise thermoregulators because they can be behaviorally passive with respect to their environment and still often achieve temperatures within their preferred range. So they don't thermoregulate because they don't have to. At high elevation, let's see what happens. Here are the temperatures measured on boulders. Here are the temperatures measured on trees. Here are the lizard body temperatures. Thermoregulators or thermoconformers, Yulia?
Regulators?
Very much so, very precise thermoregulators. Look how narrow their preferred thermal range is available in their environments. Let's zoom in onto this extremely narrow range of temperatures in the environments. One of the things we observe is that, and this is a highly scientific results, trees are cold. Trees are too cold, in fact. 95% of the temperatures that I measured on trees were simply too cold for lizards, whereas boulders were 5 to 6 times more likely to be within the preferred range for these lizards. So put differently, an enterprising lizard can at high elevation, can effectively thermoregulate simply by eschewing tree chunks and choosing to perch instead on boulders. And that's exactly what these anoles are doing. At low elevation, these anoles exhibit the arboreal niche that is typical of Anolis lizards. And at high elevation, they've almost perfectly switched over to perch on boulders. So why does this matter? Well, our entire understanding of the adaptive radiation of anoles is largely centered around the incredibly tight connection between structural microhabitat use and ecomorphology and the morphological traits that organisms exhibit. So put differently, if you alter where an anole perches in its habitat, you're probably also altering the selection it experiences on morphology. So our next step was simply to measure traits that might have shifted, morphological traits that might have shifted with respect to this change in microhabitat use. And these distill down to skull and limb dimensions. When we look at wild-caught lizards, so on this plot, I'm showing you residual head height. And so a higher value indicates a broader skull, and a smaller value indicates a population with a more dorsoventrally compressed skull. What we found just by observing, wild-caught adult lizards is that these high elevation lizards have dorsoventrally compressed skulls. They have flatter skulls. This makes sense because these lizards don't just perch on boulders, to warm up or to stay warm. They defend them as territories. They sleep under them at night. They retreat under them if they're threatened by predators. And having a more dorsoventrally compressed body eases access into the crevices that these organisms occupy. So I brought a subset of these lizards back to the lab from high and low elevation, and I just let them, I raised them under common conditions and remeasured the same morphological traits in the laboratory rear generation and found that those differences persisted. And so these high elevation lizards, this morphological difference between high and low elevation lizards has a heritable basis. So it seems to be an evolved response. We also observed that the femora and tibiae, the hind limb bones of these high elevation lizards, are also shorter with respect to their low elevation counterparts, and those differences persisted in the laboratory rear generation, so they're also heritable. And this is a slightly harder trait to explain, but there are studies showing that limb parity, that is similarity in limb dimensions between fore and hind limbs, assists with stability during climbing. And these are boulder-dwelling lizards at this point. So what's the broader point? The broader point is that any behavioral decision an organism makes likely, transcends multiple niche dimensions. So to return to this Anolis cybotes, when it's perching on a leaf, it just hasn't just made a decision about its thermal environment, it's also made a slew of decisions about its radiative environment, its hydric environment, the substrate it's perching on has a certain height, frictive coefficient, surface area. It's made a decision about, even the predators, competitors, and parasites that it's going to encounter. So it isn't even just the case that that behavior can both drive and impede evolution. In fact, it may often be the case that the same behavior that might be required to slow evolution in one dimension must do so at the expense of exposing organisms to selection in another. Okay, so at this point, I hope I've hammered in the dual role of behavior as both a motor and a brake for evolution. And I'd like to just ask, why does this matter? Well, one of the contexts in which behavioral thermoregulation most definitely matters is in the context of global change. As temperatures continue to rise, we know that climate warming presents a threat globally to biodiversity, but a growing body of theory underscores that the impacts of climate change are unequally distributed across the tree of life. Specifically, tropical organisms, especially ectotherms like anoles, should be especially vulnerable to the pernicious effects of rising temperatures. Now, organisms have the ability to combat these effects, for example, by shifting their geographic ranges upslope to track the emergence of thermally suitable habitat. Yet, summit-dwelling organisms are limited in their ability to do so before they essentially run out of room. And therefore, tropical organisms, especially ectotherms like anoles, might be riding, under climate warming, a really rapid escalator to extinction. Yet, the more I work with these high montane anoles, the more surprised by how warm I am, by how warm they like it. In just about every physiological dimension I've measured, almost every dimension, they're physiologically indistinguishable from their low elevation counterparts. And so our first step when exploring this idea was simply to ask, what's going to happen to these lizards as temperatures rise? Are they going to be pushed off the mountain, or is something else going to happen? And perhaps unsurprisingly, what we found when we did so, when we applied these biophysical models that explicitly incorporate these lizards' behavior and physiology, we find, contrary to most prediction, is that these tropical summit-dwelling ectotherms are actually going to benefit from rising temperatures. In fact, they really benefit if climate warming is severe. And the hours available for activity substantially expand. Their environments are so cold that rising temperatures essentially release them from, cold stress during certain parts of the year. And so what I've done here is clip the ranges for Anolis armori and Anolis shrivi to just show you how much hours of activity expand as the climate warms. Or another way to look at this is just to see if the maps become purpler as under different warming scenarios. Okay, but before we get too excited, there's more to consider here. We could say that based on this, that these tropical lizards might be poised to avoid the death knell of climate warming, but there's more to consider. So as I mentioned earlier, they are restricted to these high elevation monodominant pine forests, and they're replaced at low elevation where it becomes warmer and wetter, sufficiently warmer and wetter to support broadleaf vegetation, such as cloud forest. And the ecotone between the pine forest and the wetter, more mesic vegetation is also the ecotone separating Anolis shrivi and armori from their competitor, Anolis cybotes, that widespread species. So the next thing we asked was just to say, well, what's going to happen to that high elevation pine forest? So what you see here is the predicted elevation for the cloud forest plant species on Hispaniola. And what we find is that as the climate warms, high elevation environments become more climatically suitable to broadleaf vegetation. And so as a result, we predict that the high elevation pine forest, especially under severe warming, I might add, the high elevation pine forest to which these montane species are inexorably linked is probably going to constrict. Making matters worse, Anolis cybotes, a species that currently is essentially thermally excluded from their habitats, is also going to gain hours of activity in these high elevation environments that are currently only occupied by armori and shrivi. So I'm not suggesting that Anolis armori and shrivi might come into greater direct contact with cybotes, but there may be space occupancy effects that prevent armori and shrivi from expanding into different habitat types as their pine forest constricts. We know this as the boxcar effect. So what I'm suggesting is that the loss of critical macrohabitat and the potential transport of a competitor effectively pins Anolis armori and shrivi into progressively shrinking ranges, even though they are physiologically resilient to climate warming. So why does this matter? Well, this matters because we would have missed this perspective unless we explicitly incorporated the unique behavior and physiology of these montane anoles. In fact, if we perform the same exercise using standard correlative models that don't incorporate behavior and physiology, we would have come to the exact opposite conclusion, which is that they're going to lose habitat, they're going to go extinct. So in other words, we would have converged on the same answer, which is vulnerability to the extinction escalator, but for the wrong reason. Okay, so just to wrap up, I want to spend a few minutes just talking about some of the really cool things that are going on in my lab. As Wouter mentioned, we have moved to Yale recently. We are recruiting, and some of the things we're working on extend beyond anoles. I've been fascinated for a very long time by Appalachian salamanders. This is a lineage that is extremely diverse in North America, in temperate North America, whereas most herp lineages achieve peak biodiversity in the tropics. And yet, despite this incredible species diversity of Appalachian salamanders, especially woodland salamanders in the genus Plethodon, which is what I'm showing here, there's very little ecomorphological divergence, and in fact, there's sort of a classic case of non-adaptive radiation. And yet, as lungless ectotherms, their ecology is really tightly linked to temperature and moisture. So some of the things we're already finding is, sort of hidden microclimatic specialization, with some species in this lineage specialized to warmer, moister environments, and other species adapted to cooler and drier habitats. So we're sort of exploring this hidden eco-physiological dimension of the Appalachian salamander radiation. We're also exploring, other phenomena through the lens of evolutionary physiology, such as the transition to live birth. For this project, we're largely focusing on phrynosomatid lizards, because this is a lineage in which viviparity has repeatedly evolved multiple times. And what we're finding is that in every single case, the transition to live birth is accompanied by physiological cool adaptation. These viviparous lizards are cool adapted in their physiology, and they also prefer it behaviorally very cold. Even when their environment is warm, they like it very, very cool. And we show, using the metabolic theory of ecology, that this serves to metabolically compensate for the higher costs of pregnancy without precipitating evolutionary shifts in other aspects of the phenotype, like body size. We are, of course, continuing with our work on the adaptive radiation of anoles. So as I've mentioned in this talk, a lot of this story was structured around ecomorphological evolution, but evolution into distinct thermal macrohabitats and microhabitats is a relatively underexplored aspect of this radiation, and we're sort of stitching together the multidimensional story of the Anolis diversification in the Caribbean. And this is work being led by a graduate student in my lab, Brooke Bodensteiner. Finally, I just want to give one little shout out to this really interesting project I'm working on with two collaborators here at Yale, Alvaro Sanchez and Maria Rebolleda Gomez, both in EEB. And this is, we're going to try to answer some pretty interesting yet unanswered questions about how adaptive radiation proceeds. So a lot of what we do as evolutionary biologists, especially those of us working in vertebrate systems, is to sort of reconstruct what happened in the deep past. The anoles are a very old radiation, and that sort of limits our ability to sort of understand how things played out. Why is it that some axes saturate before others during adaptive radiation? Why is it that adaptive radiations tend to follow a really precise order? These are sorts of questions that we can gather pattern from, but it's much, much harder to infer process, for phenomena that occurred in the really deep past. Those constraints are released when you work on microbes, and that's what Alvaro and Maria do. And what we're doing is leveraging the extremely exquisite known genotype-phenotype map of E. coli and using evolutionary simulations to sort of reconstruct adaptive radiation in real time and ask why and how different axes saturate at different points, or just, the mechanics of how it actually goes down. How do epistasis and, genetic distance play into the timing and pattern of adaptive radiation? So these are just some of the questions we're asking, all really exciting work. And I hope I've imparted on you the really important role of behavior and evolution, and hopefully excitement for anoles as a system in general. And with that, I would love to thank you for attending the talk, even digitally, and I would be really happy to take your questions. Thank you.
Thanks so much, Martha. Super interesting work. A really complete story as well. I think we can all strive for that kind of work. I'll start with a question of my own. So one of your first results that you showed was that there's this big elevation decline in CT min, right?
Correct.
Within species as well. And so I guess an obvious question is, how much of this effect is maintained in a common garden?
That's a great question. So I don't show the data here, but I actually did bring high and low elevation lizards back to the lab, and I didn't do a common garden experiment, which would have been the ultimate test. But I did do an acclimation experiment, and a very severe acclimation experiment, actually 3 weeks long. And I was only able to drop CT min in low elevation lizards by about 1.5 degrees. So I think there's definitely a plastic component to these differences in CT min, but I don't think plasticity is sufficient to explain the very broad differences in CT min that we actually observe, across this broad climb. But it is certainly a contributing factor.
Yeah, and the skull shape differences were maintained in the lab, right?
They were maintained in the lab, although the magnitude of the effect was also a little bit less strong in the laboratory rear generation than in the wild population. So again, there might be some additional component of plasticity in modifying this.
Cool, makes sense. Would you expect evolution of movement ability or matching habitat choice? So they're matching the phenotype with the environment.
Yes. And I wish I could test it. I mean, so there are experiments in so I'm going to interpret the version of the question or offer an answer to the version of the question I think is being asked, but if I'm deviating from the person's original question, please do let me know. So one thing I've wondered is, to what extent is the perching behavior plastic between these high and low elevation lizards? Is it the case that if you moved Anolis shrivi and armori to low elevation, they would suddenly start perching on tree trunks? And if you moved Anolis cybotes and longitibialis and what have you to high elevation, would they suddenly start perching on boulders? Maybe. I haven't been able to get permission to run that experiment, but it would be an excellent one to run. But I can share that it's not just boulder perching behavior that differs, but also the relative use of sun and shade. Now, I've spoken, so the high elevation lizards are almost exclusively found in sun, not because shade isn't available, but they preferentially bask. Whereas low elevation lizards are very rarely found in open sun. They're more often along the edges of forests on the side of a tree that happens to be shaded. And shade use varies clinally with elevation, such that species in this lineage, regardless of species identity, will progressively use more and more open habitats as you go up in elevation. So there are studies on Phrynosoma, a different lineage of lizards, that showed that when you move a shade-preferring Phrynosoma from low elevation to high elevation, it will immediately seek out open habitat. Whereas if you do the opposite experiment, they will seek out shade at low elevation. So at least with respect to shade use, if you transport a lizard into a different environment, it seems to know what to do, right? So some aspects of these behaviors are relatively plastic. Perch use, I'm less certain about. I have seen low elevation cybotoids on boulders, high elevation cybotoids on tree trunks. I think this plays upon existing lability in microhabitat use. To the extent to which that's become evolutionarily fixed, I don't yet know. Sorry, that was very long-winded, but it's a question I've thought much about.
Yeah, it's not a big surprise, I guess, that behavior is more plastic than other things. So are there any differences between males and females and how they interact with the thermal environment?
We didn't explicitly test for this. In part, there's a reason for this. At high elevation, the lizards are much flightier and less abundant. And so we were unable to get sufficient sample sizes of males and females in order to statistically parse that apart. So for the purposes of the study, to ensure we had enough samples to actually run tests, we did our statistics on male lizards. That being said, I've measured lots of body temperatures from female lizards. I've observed their perch use. I don't have the statistics to support this, but to the extent to which I've been able to look at this, I haven't actually observed, appreciable differences in how they use the thermal environment. I've also measured physiological tolerances in males and females, and again, they seem to be quite similar. There's contention in the literature. Some studies often will find behavioral differences between males and females, especially depending on whether or not the female is gravid, but other studies don't. And so it's a little bit up in the air. In my system, it seems to be the case that males and females interact with the thermal environments in a similar way, though I would need to test that more concretely to be certain.
Cool. So in the scenario of warming, even though the Cytobes lizards will increase their activity and are more broadly dispersed, are the two other lizard species mentioned more specialized in their niches and able to outcompete Cytobes even with a narrower range?
It's unclear. And I don't actually think, so these lizards are parapatrically distributed with respect to each other. So I've never seen Cybotes and Arboreal or Cybotes and Shrevi interact. The few studies that have been done show, we know for a fact that Cybotes is a much bigger species, and one of the biggest determinants in contest success in anoles is body size. So just based on that metric alone, we might suggest that, that Cybotes might be a stronger competitor. But in this case, I'm not certain that the species actually directly compete for access to resources. I think where one habitat ends, where you get, broadleaf forest sort of transitioning into open monodominant pine forest, that's when Cybotes begins to be replaced by these other two species. One could make the argument that, well, okay, maybe this broadleaf forest makes its way up the mountain, and these are behaviorally labile organisms. Maybe they'll just move into the closed canopied habitat. That's where I think the difference matters, because I think the movement of broadleaf forests can actually transport Cybotes up the mountain, and the biophysical models support the idea that they can now, more easily withstand temperatures. Those environments become more thermally accessible, and if so, then they might, present a roadblock for any habitat shifts that the Montane anoles might otherwise potentially have done. It's hard for me to say without doing more competitive experiments between the organisms.
Yeah, makes sense. So interactions are always a bit more tricky.
Mm-hmm.
So maybe a bit more broadly philosophical question from me then. If we're thinking about linking macro evolution, if we think about rate of evolution, in a macro level for the Anolis, we often think about rapid speciation and this radiation. And a lot of your work is more microevolution within species where we have faster or slower evolution of traits, right? So maybe you can say something about how you think these relate to each other and whether one is necessary for the other.
I think they do relate to each other. I think the interactions that I observe at the microevolutionary level, even at the population level, create hypotheses for macroevolutionary signatures, which turn out to be borne out. So there are a lot of broad-scale phenomena that can be explained just by these, population-level studies. It's the macroevolutionary perspective provides this top-down approach, but through these, behavioral studies, mechanistic studies, we can work our way from the bottom up, and they can inform and explain each other in a way that I think would be hard to do if you didn't simultaneously consider both perspectives.
I could not agree more. So one final question then. Thus, within habitat variation in daytime temperature, if it exists like it does on Hispaniola, in mainland Anoles habitats, lead them to adopt behavioral thermoregulatory strategies like the high elevation Hispaniola Anoles favoring boulders? Or are mainland Anoles pretty much all thermoconformers tracking the thermal climate?
I don't think they all are. I don't think all island lizards are thermoregulators. I think these are general trends which seem to hold pretty well, but I don't think it's universally true. And I don't think that boulder use is exclusively the provenance of high elevation thermoregulation or cold environment thermoregulation. In fact, we have aquatic anoles, for example, Anolis Eugene Grami from Haiti and Anolis vermiculatus. Oh, no, that's not boulder dwelling. Anolis barchii. This is a species in Cuba at low elevation that perches on boulders. There are some aquatic species in the mainland that use boulders as a preferential perch type, like Anolis aquaticus, for example, in Costa Rica. So there are many reasons why organisms might switch their microhabitat use, which may or may not have anything to do with temperature. Especially if the microhabitat is in a closed canopied environment, where within that environment, a lot of the temperatures are going to be much more stable and thermally homogeneous. It's very hard to tell there. This is something we're starting to work towards, but it's going to take a lot more data, especially from the mainland, which has been, in my opinion, relatively understudied. Giancarlo Salazar from WashU is one of the few who actually systematically even got some of these data from mainland lizards. There were some studies in the 80s. It went quiet for a while, and it's starting to really pick up again. So I think in a few years, we'll be able to answer that a bit better. I will say that a lot of those mainland anoles not only live in high predator environments, but they also live in much more close canopied, typical tropical rainforest. It's not that those habitats don't exist in the islands. They certainly do. We get a lot of cloud forests and closed canopied habitat, but it might be the case that in many cases, mainland anoles are by necessity more thermoconforming. There are habitat edges, however, and breaks in the canopy that allow thermoregulation to occur, and there are species that do thermoregulate. I'm thinking off the top of my head from my own experience, for example, Anolis limifrons from Costa Rica is a species that seems to thermoregulate quite well. So I think there's going to be a lot more nuance, and I've sort of treated islands and mainland a bit more binary than they actually deserve.
Excellent, thank you. I think that's all we have time for today. So I'd like to thank Martha again for a wonderful talk and her patience with all my annoying questions.
Oh no, they were great. Thank you so much.
And I'd just like to remind you that next week we don't have a seminar. We'll have one big break, but I hope to see you all in 2 weeks. And goodbye and stay safe.